Hello students. So today we'll discuss about endospore staining. So the aim of the experiment is to perform endospore staining and to basically identify whether a given bacterium is spore forming or non-spore forming. The requirements for the experiment includes endospore forming bacterial culture, the dye malachite green, safranin, distilled water, and glass slides. The reagents that we'll be using for the experiment includes the primary stain, that is malachite green, which is prepared 0.5% weight by volume in distilled water. Then we use distilled water or tap water as a decolorizing agent, and 2.5% weight by volume alcoholic solution of safranin as a counter stain. So endospores are basically spores that are produced by the bacteria in order to endure unfavorable environmental conditions or stress conditions. So for the staining, we make use of a differential stain that is going to selectively stain the bacterial endospores. The main purpose of the staining method is basically to differentiate between bacteria that are able to form spores from those which are not able to form spores. So the endospores are basically metabolically inactive structures and they are resistant to extreme environmental conditions. The bacteria can remain in the suspended state or a dormant state until favorable conditions are present, and then they can germinate and return to their vegetative stage. Spores can be located at various positions within the cell. They can be either terminally present in the bacterial cell. Sometimes they are subterminal in position or also medial or central in position. Most of the times, the part of the cell that contains the spore, it will swell, and it is basically known as a sporangium. So in talking about the principle of the staining method, we are basically making use of a primary stain that is malachite green, which is forced into the spore wall by steaming the bacterial emulsion. Malachite green is water soluble and has low affinity for cellular material. So vegetative cells may get decolorized with water, and we make use of safranin as a counter stain that is going to stain all the cells that are decolorized or colorless. At the end, vegetative cells will appear pink in color, while endospores will appear dark green or blue-green in color. So in endospore staining, we first need to take a clean grease-free slide and make a smear of the bacterial culture using sterile techniques. We air dry and heat fix the organisms on the slide and cover the part of the slide with a blotting paper. We can then steam the slide over a we can keep the slide on a boiling water bath and add the dye malachite green and allow it to steam for at least 7 to 8 minutes. After this, we wash the slide under slow running tap water or using distilled water. Counter stain with safranin for about a minute, after which we wash the slide with tap water or distilled water, followed by blot drying or air drying. We then observe the slide under the microscope for presence of endospores. This is the diagrammatic representation of how the staining will take place at different stages during this staining procedure. So, as you can observe in this diagram, these blue-green color structures are endospores which have retained the malachite green dye, while the pink color structures are vegetative cell. Here you can see the racket-shaped structure where the spores are terminal in position in the bacterial cell, while here you can also see certain mature spores which have actually been released after the death and degradation of the vegetative cell. So our results suggest that we are able to observe endospores, which concludes that the culture is of spore-forming bacteria.